Hello everyone. Today we are going to be taking notes on something called the central dogma. So the central dogma is this idea that DNA codes for proteins and proteins result in our traits or our characteristics. So if we start with the cell, I'm not great at drawing circles. So inside of our cell, we have a nucleus. Inside of the nucleus, we have DNA, and the DNA is kind of all loose because this cell is in interphase. So we have a cell, we have the nucleus, and then we've got these things called ribosomes. And some of this should be review. So if we take our DNA and it gets all coiled up, it looks kind of like this. So this is a chromosome, and I'm going to draw its sister chromatid on this side. So these are identical, and you have in the center there, you have your centromere. So this is kind of like what we're used to looking at chromosomes. We're used to seeing this. So the chromosome is the whole strand of DNA. So if I zoom in on this strand of DNA, what I see is that it is actually a double helix. And what we have here is, if we zoom in, we've got these bases on the inside and the backbone on the outside. So there's really two strands. Now these ones are further apart, and I did that on purpose. So I want to show you how this works. So if I do some labeling here, what we have is we have the backbone. So the backbone are the two different strands that form this twisted shape. And then on the inside, we have the bases. And then I'm going to actually write some bases out here. So let's have an A, C, a G, an A, a T. Let's do another A, T, C, and G. I can put them in whatever order I want. On the other side, DNA has what we call complementary bases. So every time I have an A, I actually have a T on the other side. And every time I have a C, I have a G on the other side. So these are bases. And then a section of DNA, like say from here all the way down to the bottom, that section of DNA, we're going to call that a gene. So a gene, which this is a new vocab word. What a gene really is a gene is a section of DNA that codes for a protein. And when I say codes for, what I'm really saying here is the code is the sequence of bases. So this sequence of bases would go A, C, G, A, T, C, G. You just read one side. A different gene would have a different sequence of those bases. Now, if we look back at the beginning, DNA, what it's doing is it's coding for the protein. And then proteins cause our traits and characteristics. And when we say that DNA codes for proteins, what we mean is that we have these genes, and each gene is going to result in a different protein that is made by your cell. So it all starts with DNA. So let's look at a couple of characteristics of DNA. So the first characteristic of DNA is going to be that it is double-stranded, and it has this helix shape. And a helix is really just a type of a spiral. So we can see here that it has this sort of spiral shape. And when we say it's double-stranded, we mean that there's this one strand of DNA here, and there's another strand of DNA here. DNA is a polymer. It is made of nucleotides. So nucleotides are the monomers, and nucleotides are actually made of multiple things as well. So they're going to be made of a phosphate, a sugar, and the base. And the option for bases are A's, T's, C's, and G's. And I noted it over here. So G is always paired up with C, and A is always paired up with T. So even if I have a T here, I'm going to have an A over here. Every time I have an A, I'm going to have a T opposite it. Every time I have a C, I'm going to have a G opposite. So there's a C on this side, so G is opposite. And we call that complementary. So bases are paired and they're complementary. And so what we mean by that is A, which really stands for adenine, is always paired with thymine. And cytosine, which is C, is always paired with guanine. So that's how they're always going to work. Another thing about DNA is that the strands are held together by hydrogen bonds. So we didn't draw it on our model, but we can add it in. There are really these bonds that occur between the bases that are going to hold this together. And we're going to spend quite a lot of time on this. So we'll just draw a few of those bonds in there. And then the last thing that I want to put here is that the strands are anti-parallel. I think that's how you spell parallel. So if they're anti-parallel, what that means is that one strand points up and the other strand points down. They run in opposite directions. We're going to spend quite a bit of time on this anti-parallel nature as well. But those are sort of the main ideas about DNA that make it so that you can use it as a code in order to make proteins. And if you can use it to make proteins, then proteins result in our characteristics. And so the reason that we often say you are what your DNA is because our traits are caused by proteins, and proteins are coded for by DNA. So if we change our DNA, we change our protein, which changes our traits. And that's all for now.